Hi Hires, so Sustainability and Interdependence Unit, we're still looking at food supply, but this time we're looking at specifically exactly how photosynthesis works. This is the only other big equation apart from your respiration. Right, so overall photosynthesis, nothing complicated to be honest in terms of where we're starting. We're starting at the Nat5 level, and there's nothing wrong with this. This is a perfectly valid statement in terms of photosynthesis. So you should know already that it's a set of enzyme controlled reactions. In your head now, you should be saying, okay, well, that's got a whole pile of potential metabolic pathways involved. I could be inhibiting, I could be increasing this. And you know there's a temperature link there because enzymes and their temperature profile. And just the fact that it's fixing light energy to chemical energy, that's Nat5, but still a good start. Okay, and here is your overall equation. Again, nothing too complicated or different. I have put pigments rather than chlorophyll just because we need to talk about that one in more detail, and glucose and oxygen given out. That's fine. Okay, this is your overall summary equation for photosynthesis. Our first stage is to catch the energy from light. So let's look at light. Light is not just white light. You should remember from probably first year that the white light, if you put white light through a prism, it will split into the 7 colours in the rainbow. Okay, starting at red, running down to violet. And you might remember, and you definitely, if you're doing physics, will have a little bit of a step up on this one, that you have a longer wavelength at the red and a shorter wavelength by the violet. What this also means is that this is the most energy is in this part of it, and this has less energy. We're not going to talk about that one in particular, but the wavelength matters. Okay, right. So if we're going to look at the wavelengths, what we tend to do is to put them into a spectrum that we can measure. So we can look at it lots of different ways, and here's a spectrum with all the colours in them, and you need to know roughly these two numbers. So you need to know roughly 400 to 700 nanometers, and you need to know that's the violet end and this is the red end. Okay. If we're going to look at it in detail, then what we can look at is exactly which wavelength excites, that's the correct term, a particular pigment. So we're looking at which one actually going all the way along, starting at 400 to 401, 402, 403, all the way through, and work out exactly which wavelength the pigment was active at. Okay, so I can do it in two different ways, and you could have this in two different ways in exam questions. So we could look at it in terms of what gets blocked out. So here's another spectrum. Now this is in colour, but if it was an exam question, they obviously can't show you in colour, so it would just be a white block. Okay, and you'd be expected to understand what each bit meant. So if I shine this through a pigment and then split it with a prism, what we'd find is that some areas are blacked out, that there is no colour coming through at those points. And those bits are the bits where the pigment has actually absorbed it. Now this would be a pattern from a normal plant's kind of pigment absorption, and you'll notice that this area has not been absorbed particularly well. This one has, and this bit has, and that's what we expect. This is why plants are green, because they're not absorbing in the green, they're reflecting the green. Again, first year colour theory. So instead of looking at just in terms of blocking out, the other option is to put it onto a graph that you could work with. And what that means is that you put a graph with wavelength along the bottom, and then going up the side you have absorbance. So our first main pigment is chlorophyll A, and this is its absorption spectra, and it has some very, very clear peaks. It is very clearly absorbing in the blues and in the reds, and it is really not absorbing in your mid kind of greens and yellows. Here's chlorophyll B. So another of our main pigments, hence the fact that we normally just say chlorophyll, but now you're going to say pigments. And here we have for B, and it's a slightly different profile. So chlorophyll A had a peak here. Chlorophyll B is a little bit further in. Chlorophyll A was also peaking around here as well. So this is a kind of similar profile, but it's not exactly the same. So overall, this has maybe broadened it a little bit kind of further into this section here if I put the two of them together, which is what we're going to do in just a minute. Your third set of pigments we have here are the carotenoids. So these are accessory pigments, and you get two big groups of these, the carotenes and the xanthophylls. So if I look at these, their absorbance is even further in this way, so it's doing more absorption up here. Not really helping up here, but then your chlorophyll A and B was really good at this end. But you'll notice we still have a block in the middle, which is green, which is just not getting hit very much at all. So if I want to put all of those together, I can make something called an action spectra. So I take all of the different absorbances and I look to see actually what does it mean in reality? What do I actually get out of that? And what we're looking at here is not the absorbance, but how much photosynthesis occurs. But when you match it up, you can actually see where this pattern comes from. So here we go, photosynthetic activity, your wavelength along the bottom. And here we've got a much broader, but what we've got is the sum of all of the absorption spectra. So like these are your accessory pigments coming in over here. Here's your A and B, here's your B and A, okay? And all of these are adding up to give you a pretty decent action across lots of different parts of the spectrum. Now, that might look like a really picky thing to know, but if you're trying to actually speed up photosynthesis, this tells you exactly what wavelengths are the best ones to give plants. And that's becoming more and more of an issue if you're trying to do things like vertical farming, where you put trees basically and you use different wavelengths of light to stimulate the particular plants. And if you know exactly what the plants want, you can give them exactly what they need to get it as good as you can. And remember, this all links to food production, because this is photosynthesis, which is what is making the food. And that goes back to food security. Okay, so photolysis, which we had in Nat 5. So this is our basic. I'm going to add just a couple of things to it, and then we're going to look at it, and again, the detail you need to know at this level. So photolysis, you could split it that way in terms of the word. Photo means light, like a photograph is a picture made of light, and lysis is to split. So you're splitting with light. So this is either photolysis or light dependent stage. And we use light energy to split water apart. Now this is Nat 5. And that also gives you hydrogen for the next stage, which is essential, and you get energy out, which is ATP, which you need for the next stage, and we release some oxygen. So we take our water, we bring in our light, absorbed by, I've just put chlorophyll, but you should now recognise it will be different pigments, including chlorophyll. And then we get oxygen, which is released just by diffusion, and our hydrogen. Now, you should realise that our hydrogen is not going to kick around as hydrogen, and our energy is obviously actually going to ATP. Hydrogen is going to have to be picked up by an acceptor. Now, in respiration, you had NAD, nicotine amine adenine dinucleotide, if you want to be posh, and in photosynthesis, we have NADP, nicotine amine adenine dinucleotide phosphate. Okay, if you want to be posh, and that is converted to NADPH as its reduced form. So we are going to take from this stage our hydrogen and our energy, and that's what we're going to use in the second stage. Okay, so this is not bad, but it's not enough detail. Here's your detail. Okay, so what the light is actually doing is producing high energy electrons inside the pigment. So it's promoting electrons inside the pigments to a higher level of energy. And so what we've got now are high energy electrons. Now this should be starting to go, oh, I know what they're used for, because it's the same thing as they were in respiration. High energy electrons are used to create hydrogen ion pumps action. So they're powering them. And exactly the same way as you had in respiration, we have an electron transport chain. So inside the chloroplast, okay, inside the green sac where you've got your chlorophyll, okay, you now have pumps which have now just got a pile of energy. So they've been kind of zapped with some high energy electron, and these pumps start pumping in hydrogen ions. So we've now got an increasing hydrogen ion concentration, which you know is really useful because that can drive ATP synthase. So what we've now got is our ATP synthase, hydrogen ions going back out through this, and we're getting our ATP. excuse the slightly scribbly drawings. And we've still got our high energy electrons. Now, what we have to realise here is this is not the same as the respiration chain at this point. We're not going to use oxygen to make water. Instead, those high energy electrons which have kind of worked their way down these pumps, because remember it's a chain, when they get to the end of their chain, that's what actually breaks the water into your oxygen, which you will remove, and your hydrogen ions, really, which are going to be picked up by your NADP to make NADPH. And we now have ready to go to next stage our hydrogen and our energy. Okay, so slightly different from the electron transport chain in respiration, but not massively. Okay, so second stage. Now at NAT5, all you said was bring in the carbon dioxide, which you hadn't used already, bring in the ATP and bring in the hydrogen from the initial stage, mix it all together and you get glucose. And that was you. Okay, this is obviously far too simple for you, so we're moving it up a level. So here we go, we've got a cycle, and we're going to start with a 5-carbon molecule called ribulose bisphosphate. You need 3 of them, and you need to know the name, and you get away with RUBP though. Now, so we've got our 5 carbon molecules. We're going to bring in a 1 carbon molecule, carbon dioxide. Now I had 3 ribulose bisphosphates, so I'm going to use 3 carbon dioxides. Put them on, and a 5 plus 1 gives you 6. Hope your sums are still paying attention to this one. Now, okay, this is not very stable, so what happens is it automatically becomes 6 sets of 3. So 3 6s, 2 6 3s. Now, the red carbons are just so you can follow the carbon dioxide that's come in, but they are just carbons, so they just look exactly the same. You need to know the enzyme that's making this happen. Okay, this is Rubisco. I would just learn it as Rubisco because the real name is there, but let's face it, Rubisco is easier. Right, so this 3PG is going to be converted. We're going to need to use the energy that we brought in and some hydrogens that we brought in, and we're going to convert it into G3P, which is glyceraldehyde 3-phosphate. Now this is also a 3-carbon molecule, so we've got 6 of these. Now at this point, we can take one of these off the cycle. Now, so I've got now 5 3-carbons, so I can convert 5 3s to 3 5s. Okay, now I've just drawn a dotted line because it's several steps, but you don't need to know them all. We are going to have to use some more energy to make that happen. So if I go round twice, I can manage to get 2 of these 3 carbons. And once I've got 2 of these 3 carbons, I can make it into the 6th carbon, which is glucose. Okay. Glucose, you should know, could be used for respiration straight away, or I can convert it into starch, or I convert it into cellulose. Your new one for the higher is that it can be used for biosynthetic pathways. So that is, well, basically you now have a short section of fixed carbon, which you can use for loads of different things. You can use it to make amino acids, you can use it to make other sugars. It's there to be as a starting point. Okay, right, that is your photosynthesis. So just to have a where it's happening, this is a larger picture of a chloroplast cut away, and so what you have is a double membrane, the same as you have in the mitochondria, which is endosymbiotic theory, that at one point this was a free living thing which we've now taken over. So in the granum, so in these stacks that we have here, these little discs, each of these discs, okay, that's where you have the pigments, so that's where you do the light stage, okay, photolysis. And then all of the liquidy stuff around the outside, so all of this fluidy things that's in here, that's where you get carbon fixation. That is called the stroma. So if we want to measure photosynthesis, just to kind of clarify how we would do this overall, we have our we could look at oxygen bubbles and things like that using bubbler experiments that we had at NAT5. But what we tend to do is to measure on a large scale, we look at productivity. And so that's the rate of production that we're getting of a new material. So plants are doing photosynthesis, they're going to increase in biomass because they've got more starch, more cellulose. And we can measure that as per unit time. Here's our planet. spread out in terms of productivity. This is net primary productivity. And you can see some kind of clear areas where things are great and things are not. So obviously here, even though this is plenty warm, it's actually desert mainly, so we have very poor water availability, so that's going to impact. Around here, on our equator, we obviously have a wonderful amount of light, constant temperatures, everything's great. And then as we go further away from the equator points, you start to see it kind of dimming down, as it were, up into where we are. And that's really because we've got seasonal impacts. And then once you get up to the top end, we obviously are getting nothing because it's too cold and not enough light. So this was in the course prior to a shift, but just to kind of say potentially it could be problem solving. So we have some other terms. So assimilation is how much is actually ending up in the plant after they've used the energy that they needed for respiration. Okay. Biological yield is how much you get of a crop from a given area, and the economic is how much of the crop that was useful is coming from that particular area. And so that gives you something called the harvest index. Now, literally just showing you it, it's no longer in the course, but I could totally see it being used as a problem solving. So dry mass of the economic over dry mass of the biological. And the reason how I could see it being used as problem solving, because you have this thing about dry mass, and that is using a valid comparison because you've got no variation in water level. Okay, and that is now photosynthesis. You might need to go through that a couple of times.